Thanks everyone. I'm glad to be here to share my research work with you. Firstly, thanks for the future blood testing network on the spot for our project. So today we'll share with you about the wearable and implantable optical fiber biosensors in the healthcare and medical application. So this is an overview of this talk. So firstly, we will look at the motivation and also talk about optical fiber sensor technology. And then we'll go to this project, which will include the deliverables, milestones, and objectives, and some supporting data. And then we will talk about the next step of this after this project. And then we will, apart from that, we'll look at other projects relating to the wearable, implantable project from our group, which will firstly will be the wound care smart tracing, and the second one will be the central contraction monitor, and third one will be under the tracheal tube sensing system. Okay. So the first question always be asked is why? So why wearable? Why implantable? So I think most people have had the blood test for one reason or another, and for the most of the diseases currently, the diagnosis method is imperfect. So the monitor of the biomarkers is mainly rely on the blood or serum test in the hospital. And most of the results are slow due to the sample collecting and also transporting to a lab and tests with testing kits, for example, the ELISA test kit. And also it will come up with high cost of resources. With increased application with wearable sensing system, they will would remotely this monitoring practice. So this is a good example to show the wearable sensing system. So, for example, the sensing system can be able to sense from the human bodies all about some parameters like heart rate and blood oxygen saturation, or some respiratory rate and the temperature from breath, from humidity from breath, also some biomarkers like the glucose there. So many advertisements, for example, like the faster response and also reduced length of the hospital stay, and they can be very tiny size and real-time monitor. All right, so optical fiber. So I think most of the people know optical fiber because of the communication usage, but optical fiber can be also act as a sensing platform due to its lightweight and small dimension. So the typical diameters of the optical fiber is less than 125 micrometer, and also it's immune to the electromagnetic interference. And as you can see, the bottom right left is a SEM image for the top view of the optical fiber. You can see how small it is. So we just doing some modification of the fiber tape size with some specific coating, and then we'll be able to sensing some biomarkers there. So the working principle is quite straightforward for the optical fiber sensing system. So we got several elements. Firstly is the light source, sensing elements, optical fiber as well, and the optical detector with measure rates. So firstly, the light emitted from the light source and coupled to the optical fiber, and then we will go to the sensing elements, which is we can do some modification on the element. And then when they interact with the measure rate, so the optical signal will be changed. So we use optical detector to detect the optical change, which can be indicate the measure of the specific biomarkers. So based on the light propagation method, so the optical fiber can be classified into reflection-based and the transmission-based. So the reflection-based mainly have two different structures. One is a functional film. Another is like have a multiple reflection surface, which can generate a interferometer structure on the fab tape. And for the transmission-based, like a tape and also like long grating is different technology. So for our project, we are focused on the first one, which is a functional film. So specific for the biochemical sensor, the functional film functionalization program is the key step for the sensing. Which can improve the sensitivity and selectivity to the specific biomarker. So there's so many methods that can be done for to finish the functionalization step. So which is firstly it's layer by layer, use the electrostatic absorption, molecular imprinting, surface primary-luminescence, metal organic framework. Okay, so let's look at the project. So this project is to develop a reversible continuous IgG, which is mainly antibody from the human body, monitor with optical fiber sensor in serum, and also provide a proof of concept data and which verify the achievability of the implantable monitor. So to achieve this goal, we have 4 packages and 2 milestones and few deliverables. So the milestone 1, about 9, so 5 sensors are successfully tested in a non-blood environment, which is PBS solution, with a acceptable limit of detection and also reusability to IgG. So we finished that on time. Also the milestone 2, which is a 3 sensor tested in the blood environment, which is we tested in serum with acceptable selectivity and also reusable to IgG. And month 6, we have a new sensors produced and also sensors, and we also have time to develop a phantom, which is a tissue phantom to mimic the when we implement the fibers into the body. Also, the phantom tests have been done. So we now dropped the publication to some relevant journal. Okay, let's look at some supporting data. So the first one I want to share with you is about our nanoparticle synthesis. So after the synthesis of the nanoparticle, we coat on the surface glass or optical fiber, and that is the SEM as well. And in the zooming part, we can see how uniform the nanoparticle morphology on the surface. So the typical size of the nanoparticle is around 20 nanometers. We also be able to synthesize different size of the nanoparticles on lab. And this is a sensor fabrication step. So firstly, we have optic fiber. We immerse that into the peritoneal solution to have a hydrogenation surface. And then after that, we coat with APS, which signalization, and then able to coat with gold nanoparticles, and then MUA, CNHS be able to capture the anti-IgG, which is the final key thing for the IgG sensing. And then the sensor is ready to be tested with the IgG. So this is the experiment setup. So light source, the light transmit the coupler into the fiber coupler, and then go to the bottom and immerse it into the solution with several concentration of the IgG. And then when the sensor absorbs the IgG, the light will be changed, then the light will be detected by the spectrometer. So this is the initial experiment we have done for the gold nanoparticle coated optical fiber sensor. So this is like initial test to detect the refractive index change. which means like we'll be able to detect some specific in the next step. So this is the result. We immersed the sensors into solutions with different refract index value, and then record those spectrum. You can see the spectral change, have a very good shift, a little bit, and also the intensity well. And that is the dynamic change of the intensity at that peak. And you can clearly see we can got a response to the different refract index solutions. So after this initial test, we coated with the functional film, which is anti-IgG on the surface of the gold-coated optical fiber. And then we firstly immerse the sensor into the PBS to get the baseline. And then we immerse the sensor into different concentration of IgG solution. And then you can clearly see the step change, 10, 20, 40, 80 microgram per mil. Also, we repeat that 3 times. All the sensors have got the response. So after that, we tested, I'll see that it's the measurement of the IgG in human serum. And also we fabricated 3 sensors to test our reusability. So all the sensors immersed separately into the same concentration IgG solution in serum. And also you can see the response, even that response got decreased with the measuring round, but we still will be able to use them 3 times for all the 3 IgG sensors. And another characteristic test is the selectivity. How you gonna know that is only sensing to the IgG, not for other antibodies? So we tested we compared with IgM. As you can see, the bar chart. So the IgM response can be ignored by compared with IgG. So which is achieved the milestone 2. And also we got time to develop in the tissue phantom and follow this protocol. We're using the VPA hydrogel phantom. So as you can see upright, so that is the real hydrogel phantoms with synthesis. And then our research fund will be able to do the phantom test as well. So the phantom is here, and then we have our optical fiber in the needle. So first step is just slide the needle into the phantom, then carefully slide the fiber into the phantom. So in this, we have pure phantom and phantom with IgM for IgG. That is the compared group experiment, which is about fiber. So you can see it does response to the IgG in the phantom, which is good. And then once we change it to the IgG sensors, we can see tiny change there, so which is good. We'll be able to detect the IgG in the phantom. Okay. All right. So the next step for the project. So this is our translational path of our project. So thanks again for the future blood test network funding. And we generate a key proof of concept data. And the good news for us, we just secured the MRC-IA funding early this week. Now we are here now. And this MRC-IA funding perfectly match our ambitions to produce our preclinical development. Firstly, we will test our technology in the lab, and then fabricate some demonstration kits to engage with our end users. And in parallel, we will file the patent and also draft the technifier. And later we will apply for the MRC DPFS to conduct our clinical trials. And then if everything is all right, we probably apply for the EMA scheme as well, and also continue about the commercialization of the sensor in specific application. All right, so those all are for the project. And then we talk more projects from our group. So the first example is smart wound dressing. As you can see here, we have few small circle, and also this few ribbon fibers. So these tiny fibers is ribbon fibers, which can detect different parameters. For each fiber, we have specific field, and to be able to sense the CO2, ammonia, temperature, and humidity. So those parameters could reflect the different state of the during the wound healing. So this is a electronic unit of this project, and also the schematic diagram for the smart wound dressing. So this project has been actually MHRA approved, ethics approved. We just have done the clinical trial with a diabetic patient in the diabetic hospital. And the second one is about the placental contraction monitor. So this is one of the activity in the big multidisciplinary group. So because the placental contraction currently there's no evidence in literature about placental contraction, so we have to use some golden standard, which is MR image, to verify that our device be able to detect the placental contraction. So we're using the optical patch and electric trot to detect the contraction on the paranormal tummy. So this is phase 1, which is MR safe PCM, which is placental contraction monitor. So the aim to observe the placental contraction compared with the golden standard. After this, then this is phase 2, which is ambulatory PCM. So in this phase, the device is more small because there's no kind of MR compatibility requirements for the device. So the device can be smaller. And also we aim to detect the placental contraction in the non-clinical environment, for example, at home. So currently we have 30 months with us to do this volunteer with this. And the third example about the optical sensors, which is the optical sensor equipped smart and tracker tube. So in the mechanical ventilations, the cuff pressure play a key role. It cannot be too high or too low, and if too higher of the pressure of the cuff, they will cause the permanent damage of the trachea tissue. If the pressure is getting lower, it won't be. I mean, the liquid will get into the lungs. So we implement the optical fiber sensor in the cuff here, and to firstly measure the pressure, contact pressure, or in the trachea. Secondly, it's the CO2, which is the oxygen saturation, based on the photoprecise signals. So in the middle figures, you can see the black line shows the cuff pressure from the manometer, and that's uniform. But for our sensor and the trachea tube, we can see there is two different. It depends on the position of the contact pressure. So this quite interesting results. I want to see like the clinical may be more interesting about maybe help them to make decisions. And that is a pulse oximeter. Sorry, is a CO2 measurement based on the nanostructure tube. So we can clearly see the PBG signal, which is heartbeat signals, and then we'll be able to detect the heart rate and also the FiO2, which is oxygen saturation. Okay, so optical fiber sensor technology can be used in so many applications: drug delivery, drug detection, medical sensing, antigen antibody, or portable monitor, implanted monitor. Some point of care application, for example, biomarkers like CO2, ammonia, humidity, temperature. So the potential application of optical fiber sensor will fit the gap between the clinical challenging and technology imitation. And the final goal is to make people's life easy. Okay, that's my thank you.